In the current medical landscape, stem cells have the potential as a solution for a host of conditions which had previously been viewed as untreatable. Specifically, because of their regenerative properties, stem cells may hold the key to treating neurodegenerative and traumatic neurological injuries. The central nervous system includes the brain and spinal cord, which allows us to move, think, and process our environment. In order to perform all these functions, it has highly specialized neuronal cells, which allow for the generation and transmission of electrical signals. Because the central nervous system is so vital for so many functions in the body, if parts of the central nervous system are damaged as a result of disease or trauma, the results can be devastating, leading to serious physical and mental disability or even death. Unfortunately, the central nervous system lacks the capacity for self-repair and regeneration, meaning that when neuronal cells are lost or damaged, they're gone for good, and there is almost no hope for recovery or return of function. This is where stem cells come in as an exciting new avenue for possibly repairing and replacing these lost brain cells. Stem cells have three general characteristics which make them ideal for repairing damaged neuronal cells. Firstly, they are unspecialized and don't have any specific structures in order to perform specialized functions, making them a blank slate. Second, although they are unspecialized, they have the capacity to develop structures in order to become specialized and perform a particular function. And finally, stem cells are capable of dividing and renewing themselves over extended periods of time, allowing for the proliferation of even more cells. Differentiation of stem cells is the process whereby an unspecialized stem cell will become specialized in order to perform a new function. What a stem cell will differentiate into is determined by their environment, most notably by the cytokines called growth factors. Growth factors are diffusible signaling proteins which act to stimulate tissue growth and repair, and are secreted by the surrounding cells. Growth factors work by specially binding to receptors in stem cells, activating associated downstream signaling pathways, which will regulate the gene transcription in the nucleus and stimulate a biological response in the cell, leading to the proliferation and specialization of that cell. What a cell will differentiate into is determined by what kind of growth factor they are exposed to. When discussing neuronal cells, there are a particular class of growth factors called neutrophins, which are synthesized and secreted in the brain that focus on the regulation of neurons in particular. Sadly, it isn't as easy as jabbing someone with any old stem cells and away we go. There are many variables that need to be investigated before this can be seen as a viable treatment. The type of cell used in the treatment has been theorized to play a role in its efficacy. Neural stem cells are immature cells harvested from the human nervous system with the capacity to differentiate into more specialized cells in the central nervous system. The sources of these cells can be embryonic, neonatal, or adult. These cells are able to take on properties which are brain region specific, meaning they are a possible treatment for a myriad of complex conditions. One such condition is Parkinson's disease, which is characterized by the gradual degradation of dopaminergic neurons in the basal ganglia of the brain, creating a dopamine deficit which causes serious motor dysfunction. Neural stem cells have been shown to regenerate the specialized cells, relieving symptoms of Parkinson's. One issue with using stem cells from an extraneous source is the possibility of rejection or long-term abnormalities such as tumors. In Aramagiglio et al. 2009, a patient who had been receiving neural stem cell therapy for 4 years developed a brain tumor, which researchers concluded was derived from the donor cells. Researchers have also been able to culture stem cells in the lab, which has two key benefits. One, they don't need to harvest new cells all the time, and two, they are able to curate the cells with various growth factors to differentiate for their specific requirements. Bone marrow is another interesting source for stem cells, because it can bypass many of the safety concerns around rejection of the implant, because a patient can be their own donor. In animal models, marrow stem cells have shown the ability to become myelin cells, indicating they have promised to treat multiple sclerosis without complications as a result of the implant. Stem cell research still has many new avenues for further study. For example, whether the cells should be implanted either directly into the patient's brain or instead through the vasiculture remains a point of contention. Obviously, if it is a viable option, it would always be preferable to use the vasiculture to reduce the risk and expertise required. However, there are concerns that the number of cells that would reach the targeted area would be insufficient to improve function. The timing of the transplant in relation to the occurrence of the injury itself may also play a role in the effectiveness of certain stem cells. It's thought that it would be preferred that the treatment take place as soon after the injury as possible, especially when the body is still naturally releasing the particular neurotrophics associated with regulation and repair as a result of the trauma. But in the case of patients with spinal cord injuries, strokes, or cerebral palsy who may have had their condition for an extended period of time, we aren't sure what effect this might have. One major limitation of stem cell research is the use of animal models for testing, which limits the applicability of the results for humans. In conclusion, stem cell therapy may hold the key to treating neurological disorders. However, it is not ready yet and needs extensive research before it can be seen as a viable treatment.